hosting today with Rick Cooley from Oregon State University. The webinar takes place every third Thursday of the month and features investigators that are using genetic code expansion to solve biological questions. It also features new amino acid chemistries and everything in between. So if you are someone that would like to share their work, or you know someone that has really interesting work that's going on, please let us know and we can try to get you to give a talk on this webinar. It's also a really good format for investigators at all stages of their career. And so we'd like to feature investigators that are early stage investigators to help advertise their research when they're out looking for jobs, or they just want to show the world all the exciting work that they're doing. And actually today we're featuring Carly Shizzle, who is on a postdoc at Alana Shepard's lab, who's on the job market. So anyone who sees this, she has really exciting research going on. So today there's two exciting talks. I already told you about Carly Shizzle, but also Linda Kenney. I believe Linda's going first. So Linda is joining us from University of Texas Medical Branch in Galveston, Texas, where they are a full professor in the Department of Biochemistry and Molecular Biology and Microbiology and Immunology. They received their BS from the University of Iowa, go Hawks, in zoology, and then went on to do their PhD at the University of Pennsylvania, studying characterization of potassium transport by the sodium pump in human red blood cells. And looking through her CV, I was excited to see that she published 3 amazing papers with one of the greats, Clara Francini Armstrong, to determine the structure of triads in skeletal muscles using electron microscopy. And I just found out that these 3 papers just came from a rotation in her lab. So quite prolific, even during a short rotation, can publish 3 unbelievably impactful studies. I went on to do a postdoc at Yale to develop an expression system for sodium potassium ATPase in yeast, and then to Princeton to study porin gene expression in E. coli. Got to start as an independent investigator at OHSU, and then moved on to the University of Illinois, Chicago, where they recently have been recruited to UTMB in 2019. I would say it's difficult to put Linda's research interests into a box, and they made several really impactful findings along their career. But the website says super resolution imaging in bacteria and single cell analysis, and the development of oncolytic potential of Salmonella for targeting tumors. Studying signal transduction in bacteria with a highlight of mechanotransduction and osmotic signaling in E. coli, which is super interesting. She's won several awards for her work, including being elected into the American Academy of Microbiology, and recently won the Texas Star Award. Could basically go on forever about Linda, but we'll get going with the title of her talk: Genetic Code Expansion Enables Visualization of Salmonella Type 3 Secretion System Components, Secreted Effectors. Linda, take it away.
Great. Thank you so much for that kind introduction. It's great to have a connection from the past with Clara. She was really had a special influence on my career. And I'm coming to you today remotely from my home in Santa Fe, New Mexico. And here is just a recent photo I took of a sunset over the Jemez from my backyard. And so the work I want to tell you about today is work that started by a spectacular postdoc, Karen Singh, and I'll tell you more about that, but started when we were at the Mechanobiology Institute at the National University of Singapore, and continued in the biochemistry department at UTMB. Okay, so let me briefly just outline what I want to tell you about today. This is an application of GCE. So I want to describe just a brief overview of Salmonella pathogenesis for the purposes of understanding this talk. And then I'll show you how we image Salmonella infections. And I'm going to use HeLa cells as an example, but we also image them in zebrafish, in nematodes, and in chick embryos, for example. And how we've used genetic code expansion to image non-abundant and impossible to label proteins. And in particular, I'm going to talk about 3 proteins. One is SifA, and that stands for Salmonella-induced filament A. And another is SSEJ, which is Salmonella-secreted effector J. And these are both virulence factors that are secreted. We also refer to them as effectors. And I also want to talk about SsaP for Salmonella secretory apparatus. And it is a small protein that is involved in the switch between secreting effectors that form the apparatus, and then the switch happens to secreting the virulence factors into the host. Okay, so just briefly then, we get salmonella infections from ingestion of contaminated food or water, and most of the bacteria are killed by this extreme acid pH of the stomach. But the few that survive then go on to the intestine. And salmonella, through signal transduction, recognizes that it's in a host environment and catalyzes the expression of what we call a type 3 secretion system. And that rearranges host actin on the epithelial cells and drives the uptake of salmonella across the intestinal lumen into the cells. And then once inside, again, salmonella resides in a vacuole, and it then senses the acid pH of that vacuole and turns on another type 3 secretion system that then modifies the vacuole so it's not degraded by the normal endocytic pathway. And these type 3 secretion systems are encoded on pathogenicity islands, which were identified when people began sequencing bacterial genomes. And these islands were acquired during the evolution of Salmonella as a pathogen, and they have a lower GC content than the rest of the chromosome. Okay, so these injectisomes or type 3 secretion systems are these incredible nano machines that are structurally homologous to the bacterial flagellar system. So there's many different components that make up this system, and their effectors then get secreted through this apparatus into the host. And our work has really focused a lot on how the bacteria senses the acid environment of the vacuole. We're focused on the type 3 secretion system encoded on SPI2. And what we've shown is that bacteria, or Salmonella in particular, unlike eukaryotic cells, can actually acidify in response to the acid pH of the vacuole. And we put a FRET DNA biosensor inside the bacteria and measured the pH when it was in a macrophage vacuole. And the bacteria acidified to pH 5.6. And they use that acid pH to then drive expression of a key regulatory protein, SSRB, that then turns on all of these genes that will encode for the secretory apparatus, as well as the effectors that get secreted. And what we recently showed is this regulator is not only upregulated threefold in acid stress, but its DNA binding properties are stimulated 30-fold by acid pH. And so that then turns on this system in the vacuole. And the challenge we have of studying this SPI2 type 3 secretion system is shown here on this slide. So we can determine whether the bacteria are making injectisomes because the surface protein, which is normally embedded in the vacuolar membrane, it's called the translocon. We purified that protein and we raised antibodies to it. And then when we have fluorescent secondary antibodies, we can see that even though all the cells are acid-induced, only about 30% of them actually go on to assemble an injectisome on their surface, right? And that heterogeneity is something that we don't fully understand. It's in part stochastic, but also in part regulated, but it makes it really challenging. Whereas the SPI1 type 3 secretion system has at least 80 copies per cell, and we have cryo-EM structures of that system. Okay, so here's what an image of Salmonella infection looks like in HeLa cells. This is a 3D super resolution SIM image. So here we have the HeLa nuclei in DAPI stained here in blue, and the bacteria are in turquoise. And then LAMP1 positive endosomes are shown here in red. And as a consequence of the Salmonella infection, what you see is the formation of these endosomal tubules. So the bacteria are co-opting the endosomes and forming these tubules that then provide membrane area for the expanding Salmonella containing vacuole, which is right here. So once inside this compartment, the bacteria modify the vacuole so it's not degraded by the normal endocytic pathway, and they replicate in this compartment and then disseminate to distal sites such as the liver and spleen. Okay, so the two proteins I want to focus on for the moment are SSEJ and SifA for salmonella-induced filaments. And they're secreted once salmonella is in the vacuole, and they get out into the host cytosol, and they have a sort of yin and yang effect on helping to form these tubules, which they also interact with the motor protein kinesin in order to generate force. So the problem with imaging these effectors, so we wanted to study them as they were in their native copy number, expressed behind their native promoters, and as they were secreted through the type 3 secretion system. So normally we had made photoactivatable fusion proteins, but you can't study these because they actually jam the secretory apparatus, so they're not secreted. So the way we got around this problem was to make HA tags, which were small enough to be secreted, and then we could use antibodies and fluorescent secondary antibodies and then image them in super resolution. And so one of our typical images is shown here, where you see here's a salmonella containing vacuole. The LAMP1 endosomes are shown in green, and SSEJ is shown here in red. In these blow-up images, you can see that SSEJ is decorating the outside surface of this tubule at 12 hours post-infection. And when we go to even longer time points at 16 hours, you see that we've almost all of the endosomes in the cell are being co-opted and used up and incorporated into these tubules. And at later times, these single membrane tubules actually form a double membrane tubule that encases cytoplasmic elements such as the cytoskeleton and other things, which are believed to help provide nutrients, et cetera, for the bacteria that are replicating in the vacuole. So we could image SSEJ really well, but whatever we did, we could not label SifA. And we put tags at the N terminus and at the C terminus, and we had a crystal structure of SifA. So we could actually incorporate tags in what we thought were surface exposed loops, et cetera. And we raised several different antibodies, and nothing worked for us to be able to label SifA. So, you know, because I'm speaking in the GCE for all that, of course, the solution is GCE to the rescue. And so we applied genetic code expansion, and this was work all done by Kiran Singh, who was a chemist by training at the Indian Institute of Technology in Delhi. And so at the time, this is about 2017 when he started, we really didn't know a lot about sort of the codon context, et cetera. So he made single mutations in the SifA gene, which he cloned on a plasmid, and that's inside the bacterial cells. And then he introduced stop codons. And he tried about 15 different positions, hoping one of them would be amenable to labeling. And several of them did indeed work. So then the bacteria are also expressing a plasmid that contains the orthogonal tRNA and the synthetase. And so then we make SifA, and the stop codon is suppressed and replaced with an unnatural amino acid, a non-coding amino acid. And then the bacteria secrete SifA through the type 3 secretion system, and then we label the SifA in the host cytosol using click chemistry. And so then what was important for labeling other proteins that were not very abundant. So this worked for SifA, but it's fairly inefficient. And then through continued work in this area by coworkers that I want to acknowledge here, they made different plasmids in which the tRNAs were evolved to be more precisely recognizing that stop codon. In particular, this 3 amino acid combination works really well. And so we then now use this for establishing our mutants for site-specific labeling. And it worked even for proteins that were not abundant, as I'll show you. Now, I just want to mention briefly, reviewers were very concerned about off-target labeling. And so when we looked at the Salmonella genome, what we see is about almost 10% of the genome uses the TAG stop codons. But in fact, only a very few of the pathogenicity island genes actually use TAG stop codons. And so one way we get around reducing off-target labeling is that we're looking at these proteins only under very precise inducing conditions. Particularly, we use acid stress. So for example, the SPY1 genes are not expressed under these conditions. And in addition, this new tRNA that we're using, its preferred sequence is here, and that's really absent in most of the other places where the stop codons are present. In addition, the TAG codons tend to be in genes that are not abundantly expressed. And finally, other coworkers established in E. coli, they actually made substitutions of all the TAG stop codons to TAA, and it really didn't influence their background labeling. So the bottom line here is that off-target labeling was really not a problem for us. And you can see in the slide now where we see the actual labeling that in the absence of the non-coding amino acid, azidophenylalanine, you see that there's really no background labeling. It's really, really blank. And here we're labeling SifA now for the first time ever because we're using genetic code expansion. Now, we really don't see the product on SDS page until we actually then look at the fluorescent labeling when we have both the fluorophore present as well as the unnatural amino acid. And then we could actually visualize SifA in HeLa cells and show that it co-localizes with LAMP1. This is just to show you the DAPI stained nuclei of HeLa cells. Here's the SifA that we were now able to image and the merged image. And in the absence of the non-coding amino acid, again, there's essentially no background. And when we then label LAMP, you can see that SifA is co-localizing with LAMP1. So we were able to show that very precisely using genetic code expansion. And Karen then went on to show that we could also avoid the click reaction, which really wasn't a problem for us, but we could use fluorescently labeled non-coding amino acids. In this particular case, we're labeling SifA with hydroxycoumarin ethyl glycine. And the SSEJ we have here is HA tagged and using immunofluorescence. And when we merge the two, you can see that SifA not only is localizing with LAMP, but it's co-localizing with SSEJ as well. And then I want to describe how wonderful genetic code expansion is for labeling a very difficult non-abundant protein called SsaP. So SsaP is a small protein, and it's 124 amino acids. We made a photoactivatable fusion to photoactivatable M-cherry, and the fusion protein was cleaved, so we could not follow SsaP. And it's part of what we call the substrate specificity switch. So normally, you first assemble the injectisome, and then something happens with SsaP that we don't understand. But then the injectisome, it switches to, instead of secreting the components of the injectisome, you start secreting the effectors that will be secreted through it. Right, so we really wanted to see SsaP and try to understand the function of this protein. And so again, we used genetic code expansion, and you can see here that you can't see it on SDS page, but we were able to detect a very diffuse band after labeling with silicone rhodamine tetrazine. Okay, so you can see in this image, again, in the absence of the unnatural amino acid, there's no background, and it's very nicely labeled using genetic code expansion. And if you look at this blow up here, you can sort of see the outline of the cell, of the bacterial cell, and you can see that SsaP is polarly localized, which we were really happy to see because we had already seen that the injectisome is polarly localized. So it's not surprising that we would find SsaP in this location. And then we could see, again, in the absence of the unnatural amino acid, you do not get labeling. And when we have an SsaP null strain, again, there's no background labeling. And now let me see if I can blow this top image up. But what we were able to see here is that, so here we have SsaP labeled. We have the bacteria labeled with LPS staining here in red. In this image, LAMP1 is green and the DAPI nuclei are in blue. And you can see, so this is 16 hours post-infection, that SsaP is no longer associated with the bacteria because it's been secreted. And we could never detect this by any other means. So we really were able to label these proteins and follow them using super resolution imaging because of genetic code expansion. And now let me go on to the next slide here. And then during COVID, our colleagues, we were hoping that we could then use genetic code expansion to label coronavirus. And so our neighbor, Pei-Ying Shi, who's a virologist, was working with Zika virus. So we decided to use that as a test case. And so we labeled the Zika virus coat protein using genetic code expansion. And we can clearly see these fluorescently labeled viral particles in these viro cells. And we could then harvest the fluorescently labeled Zika virus and then infect naive viro cells and watch viral uptake as we lose fluorescence over time. And unfortunately, Pei-Ying left UTMB, and so we actually didn't get around to studying coronavirus. But it just highlighted, again, now the ease of the technology. That first SifA labeling that Herein did using just the TAG codon took about 2 years of work. And now we can go in a couple of months to site-specifically labeling our proteins of interest. And I think now, too, with the expanding tools that are highlighted in the GCE for All, it's going to make it so much more accessible. And many of my colleagues told me when we were doing this project was, well, you know, Linda, we tried to do this too, but we couldn't get it to work. And I think now that people are going to be able to get it to work. And so lastly, let me just leave you with this image that really nicely highlights the superior specificity of GCE labeling over immunofluorescence. So in this case, we took SSEJ, which we had already labeled with using an HA tag and used immunofluorescence. And we introduced the stop codon here. And then when we label site specifically, the SSEJ, you see this really nice imaging in HeLa cells. And here we label using immunofluorescence, labeling the HA tag. And you can nicely see, this image here, that all of this background staining that you see in the merged image, resulting from the immunofluorescence, like here, that you absolutely are lacking in the GCE labeled strain. So it's just really a fantastic way, especially if you're interested in co-localization, for example, and other questions. And so let me just briefly summarize by saying that we were able to successfully label difficult to label proteins such as SifA and non-abundant proteins like SsaP. And you can avoid the click reaction if you need by using fluorescent non-coding amino acids. I showed you that really off-target labeling is not a problem. I didn't show you this data in the interest of time, but in our paper, which is here, we showed that expressing the plasmid that encodes the tRNA and the synthetase does not affect bacterial growth, and it did not impair pathogenesis. But I do want to make this one point that Karen wanted me to point out. He also used this non-coding amino acid, acridonylalanine, and that caused a cell division defect in Salmonella. So what we see is that the bacteria fail to divide and they form these long chains. So we did not use that in our studies for obvious reasons. So it's just important to check these basic parameters just to make sure that the probes aren't toxic. And finally, then, let me just acknowledge Karen here, who is just the star of this story that really got this working, and these funding sources over the years, and other colleagues who provided us with plasmids that we use for labeling. And thank you very much for your attention.
That was great. Thank you very much. We're taking questions on chat, in the Q&A, all of the things. So, or you can ask a question online. Ryan.
Absolutely, Linda. Wonderful, wonderful talk here. And I'm very excited about your imaging work. Just a quick general question on the imaging. I know the field here is just so challenging with regard to where the background signal comes from, right? And how one mitigates that. And you obviously touched on that quite a bit. You have experience with a number of different labeling chemistries, as well as the differences in dyes, and all of these things play a big factor. The two areas that we see labeling background signal come from is, some of it's from the GCE with other codons, like you mentioned, the other natural codons. And the other side is just the background that you get from the dye and the concentration that you have to use and the labeling time that comes from the dye that you throw at the cells. In your experience, you showed beautiful images that didn't have any background. What's the greatest sort of challenge right now? Is it the dye? Is it the background signal that you get from the GCE part, or is it the background that you get from the dye and the concentration that you have to use to get the labeling to happen? Where's the threshold, or is there one chemistry that you found is just better than another to reduce background?
Well, I would say that now we favor this silicone rhodamine tetrazine. So typically we are labeling somewhere in the order of 1.5 to 2.5 micromolar.
Okay.
And we label for about 2 hours, or 1.5 to 2 hours. And then Karen mentioned that he extensively washes afterwards.
Okay.
So, and he has, we have a Journal of Visual Experiments video where he's gone through those steps in really exquisite detail. But I think I don't think that off-target labeling was a problem for us, especially now that we're using the plasmid that really has the codon context of the adjacent amino acids, because that particular combination is even more rare. So I don't believe that the most of the problem is labeling other stop codons. I think it's in the dyes, and choosing those carefully and being really compulsive about washing steps is really critical.
Great. Thank you so much.
Thank you.
Yeah, the context thing is really interesting to me. This is something that I'm studying quite a bit. I think that kind of continue on with what Ryan was mentioning. It's just like dyes being stuck in lipids. And so the washing steps basically allows those to be removed that are not incorporated, right? I think that that's kind of what most people struggle with, but you obviously are not, which is impressive.
Right. Well, I think it really helped having a chemist postdoc. And, you know, because one, he's really good at trying 50 reagents and finding the one that really works well, you know. So I just want to say, I can't say enough about Herein and, you know, his ability to really get in there and empirically figure it out.
There is a question. Okay. Question from Emerson. Thank you for the presentation. Could the background of immunofluorescence be due to truncated proteins or near cognate suppression? Is the question.
You know, that's an interesting question. I just think that perhaps maybe we're not washing well enough when we do the immunofluorescence. How good is the HA tag? Maybe they're better and the antibody is binding to other things, perhaps. I don't have a good answer for that, but I do think that image is pretty, you know, stark in terms of, you know, like we're looking for interaction of SifA with kinesin. Kinesin's everywhere in the host cytosol, right? So looking at co-localization with GCE is just so much better than with immunofluorescence, right? So, and now I think that it's becoming so much more feasible that, you know, I just think, why wouldn't you do that instead?
Great.